In the name of Allah, the most merciful and the most beneficent. Assalamu alaikum. How are you all? I hope you all are feeling well at your home. Dear students, I welcome you in Pakistan International School TIES virtual learning classes. And this class is for biology for students of first year. Dear students, today we will talk about some more important points and some more important concepts about prokaryotes. In prokaryotes, we are studying bacteria in chapter number 6. And in that chapter, today we will see modes of nutrition in bacteria and growth and reproduction. So we will see first of all modes of nutrition in bacteria. So if we see mode of nutrition in bacteria, there are like in this we will study about the feeding habits of bacteria, like what are the basic methods of obtaining energy and carbon. There are of two types, heterotrophic and autotrophic. Heterotrophic means they take in food from outside, and autotrophic means they make food in the body. In autotrophic, we will see photoautotrophic, or they are also said to be photosynthetic bacteria, and then we will see chemosynthetic, or they can also be said chemoautotrophic bacteria. And then we will see heterotrophic bacteria, in which we will talk about saprophytic bacteria and parasitic bacteria. First of all, we will see autotrophic bacteria. Auto means by themselves, and troph is the level of feeding, the feeding habit. So they are generally called photosynthetic bacteria also. But autotrophic bacteria are of two types, photoautotrophs or chemoautotrophs. So autotrophs are some bacteria which are able to synthesize organic compounds necessary for their structure and metabolism from simple inorganic compounds by themselves, and that's why they are called autotrophic bacteria. And then they are of two types, photosynthetic and chemosynthetic bacteria. So photosynthetic can also be said to be photosynthetic autotrophs, and chemosynthetic bacteria can also be said to be non-photosynthetic autotrophs or chemoautotrophic bacteria. Photosynthetic bacteria, actually, they include all three coxal, bacillary, and spiral species of bacteria. So in that, all are anaerobes which grow in light and are usually found in sulfur springs where hydrogen sulfide is normally present. So very important, two examples of photosynthetic bacteria are purple sulfur bacteria and green sulfur bacteria. So these bacteria, they are like all type of bacteria except cyanobacteria, and they are having unique type of chlorophyll that is called bacteriochlorophyll, because that is especially present in bacteria. So that is different from that is different from the chlorophyll which is present in the plants. So bacteriochlorophyll. And the chlorophyll is incorporated in the membranes of their mesosomes. So we have seen these mesosomes before in the first chapter, And unlike green plants, algae and cyanobacteria, they do not use water as a reducing agent, but uh, and so they do not produce oxygen. So instead, they use hydrogen sulfide. If we can see this one, hydrogen sulfide is utilized, and in the presence of carbon dioxide, light, it forms carbohydrates, and then it does not give up oxygen. It gives off sulfur as well as water. So they also, if we talk about purple and green sulfur bacteria, so purple bacteria are having carotenoids, which are the photosynthetic pigments, and green sulfur bacteria, because that is green, so it is having special bacteriochlorophyll. And the pigments under the microscope appear to be uniformly distributed in all of the cytoplasm. So hydrogen combines with uh, the oxygen, and form of water, as well as sulfur is like in the reduced form. It just comes out, as well as uh, carbohydrates are formed. In chemosynthetic bacteria, they are non-photosynthetic autotrophs. So non-photosynthetic means they do not prepare their food while this photosynthetic process, but they are using some other inorganic substances for the production of energy for different purposes or for the food synthesis. So they are also nitrifying bacteria. They lack pigment. They don't have pigments in their bacterial chlorophyll or carotenoids, but they get energy for food synthesis by the oxidation of certain inorganic substances like ammonia, like nitrates, and like ferrous iron. And they are also using um, hydrogen sulfide in the presence of oxygen if they are aerobic bacteria, so they form water, sulfur, and energy. And if they are doing the photosynthesis in place of this carbon oxygen, carbon dioxide is added, so they form carbohydrates, then water and sulfur. Now we will see heterotrophic bacteria. Heterotrophic are the group of those microorganisms or those bacteria which use organic carbon as a food, as opposed to autotrophs like algae, which use sunlight. That means that these are these are the bacteria which do not make their own food, but they use the food from other organisms. And they found in every type of places in the water, and like we can say like we can detect heterotrophic bacteria from the water by using a method that heterotrophic plate count. So we count over there like the film of heterotrophs which are made over there. In heterotroph bacteria, we will see two types of bacteria: saprophytic bacteria and parasitic bacteria. They are having great difference. Saprotrophic bacteria, they grow in the dead, decaying organic material and live by digesting and absorbing that type of material. So while obtaining food for themselves, saprophytic bacteria gradually break down complex organic compounds into simpler products, and by secreting their enzymes to break down the carbohydrates. So that process is also called fermentation, as we have seen fermentation in chapter number 4. Its example is Azobacter, that this is saprophytic bacteria and it just lives on the deadly material. dead material. Parasitic bacteria, we know them very well. They live on or within the living organisms, both in plants and animals. So they obtain the organic food from the host directly, and they grow also over there. So parasitic bacteria, including pathogenic bacteria, which always just cause diseases, or they act as the lethal, or we can say very harmful for the living organisms. Like they cause lots of diseases in human beings and other animals also, like streptococcus pneumoniae. So it causes a pneumonia in living organisms, especially in human beings. Our next topic is growth and reproduction in bacteria. So first of all, we will talk about growth, because growth is different than reproduction. So in growth, we will see that bacterial growth is regulated by nutritional environment. If there is a healthy environment for bacteria, so they grow very well. And in the time, the suitable environment is there, that the time bacteria is incubated, its growth leads to increase in number of cells, which allow definite course. Like before, they are going through unfavorable conditions, so they are in incubation period, but in suitable conditions, then they grow rapidly. So we will see the growth curve also, in which we will see the four phases of the growth. First is lag phase, other is log phase, then we'll see stationary phase and decline phase. In log phase, that is also called logarithm or exponential phase, because here is the reproductive phase, but that is, we will see the increase in the population phase. So first of all, we will see lag phase. Bacteria adapt themselves to growth conditions. It means it needs that period that individual bacteria are maturing. They are not dividing. So they are, what they are doing, they are just maturing. It means they are increasing in size. They are synthesizing RNA. They are they are making or synthesizing their enzymes and molecules in the body, so that they can adopt the external environment. So that lag phase is just, it is from 1 hour to 4 hours. They are just going increase their size. They are not reproducing here, just to prepare themselves, and producing lots of chemicals in them, like we are having interphase during the mitotic phase. So length of this phase depend on the type of bacterial species, cultural medium, and environmental factors also. So if they are having proper environment, nutritional environment, some bacterial species, and cultural medium, what type of culture, in vitro condition they are. So then they just go through this process. Here, if we here you can see uh, here you can see the growth curve or cycle. In that we can see that there is 4 phases, and if we just see these 4 phases, the first one is lag phase, the other one is log phase, stationary phase, and death phase. So in lag phase, we can see that lag phase is just a straight line here. There is no increase in bacterial number, and according to the time of 1 to 4 hours, they are just going to increase their size. If we just talk about log phase, the second phase of that one, that is exponential phase. But exponentially, they are increasing in their numbers. So in log phase are also called logarithm phase. That's why it's called log phase. Exponential phase because they are growing and increasing in size exponentially. So the cell is doubling here. The cell is doubling here. It means the number of new bacteria are appearing per unit time is proportional to the present population. The growth is not limited, but doubling will continue at the constant rate. So both the numbers of the cells and the rate of population is also increasing and doubles with each consecutive time period is passing on. So we can plot the graph, the natural logarithm, like the number of increase in the cell or population and the time is passing on. So we can see that just giving the slope of increasing the number or specific growth rate of the organism. So the actual rate of this depends upon the growth conditions which affect frequency of the cell division events. Like if it is having the proper surviving conditions over there, they are having a proper medium, nutritional medium over there. So then it will increase more and more in number. So if we just see under controlled conditions, cyanobacteria can double their population 4 times per day. So again, in this growth curve, we can see the log phase. This is the log phase, this blue line, which is the curve line moving up. It means the number of the bacteria are increasing in this phase of their life. So here it means there is more reproduction and the population is increasing. Now we'll see stationary phase. It is few hours to days. So in stationary phase, that is due to the growth limiting factor. Like if the depletion of the nutrition or formation of inhibitory products, like some waste material, toxic substances are just producing in that phase. So that the death of the cells starts. So we can see in this curve that there is a straight line at a like specific height, means here the cells are continuously growing, no death and no decreasing of that occurs. But here at this phase that is stationary means as many cells are going to increase and divide or newly formed cells per time. So dying cells per time is the same ratio. So both are dying and newly formed cells both are having the same rate at the time. So if the substrate is depleting continuously, so here we can see the final decline phase. In that few hours to few days, it can be in decline phase. We can say the death phase, in which now the all type of nutrients are going to deplete, and the cells now they are continuously decreasing. It means the cells are dying. So that the lag phase of lag, not actually lag, that is decline phase, that's brought by exhaustion of nutrients, accumulation of toxic products, and autolytic enzymes are also there which just destroy these bacteria by themselves. So these four phases, like lag phase, in which there is growth, means growth in size and synthesis of chemicals and enzymes are there. Then there is reproduction phase, or we can say the increase in number and population exponentially. Then there is stationary phase, in which newly formed cells as well as the dead cells, they are in the same number. Then due to depletion of the nutrients, cells are dying more as compared to they are forming or they are dividing. So here we can say now the decline of this um, this phase properly. So the cells are dying completely. Now we will see reproduction in bacteria. Reproduction in bacteria, there are of two types, as we know, asexual reproduction and sexual reproduction. Right now we are going to study about asexual reproduction. In that we will see very common and very simple step of that, that is binary fission in bacteria. First of all, in that, what happened in one cell in under favorable conditions, the cell replicates its DNA, like DNA that is called nucleoid, and that DNA is replicated here, means it makes its copy. When it makes its copy, the cytoplasmic membrane elongates more from the center and separating the DNA aside. When they are separated properly, the cross wall forms and membrane invaginate. When the invagination is going to be deeper and deeper, so cross walls formed completely and the two cells are divided. So that is like they call it binary fission, because that divides into two cells. So bi means two, so fission means breakdown, so they break down into two cells. That's why it's called binary fission in bacteria. There is another process of asexual reproduction in bacteria, that is budding. And in budding means from old cell a new bud is forming, and that just makes after some time a new organism that sheds off from that old bacteria to form the new organism. So we can see here cell genetic information is duplicated first of all in every cell. This is going to be happen to just duplicate its DNA, then creating an identical copy of the clone of the original cell is formed, like this cytoplasm. A cytoplasm of this bacteria just going to be elongate and move towards another side, and around that cell membrane and cell wall appears. And when they going to appear, they are elongated too much that this DNA is completely divided from the center, so it just moves towards the other side. When it completely moved outside, so that bud appears, and later on this bud sheds out into an organism. So from here the bud is shed off, that's why it's from the scar on the old cell. And you can see here from one cell the other cell is formed, the other from other cell the one more new cell is formed. Now we will see sexual reproduction. Is there sexual reproduction in microorganism? Yes, but there are very simple procedures, not completely fusion of we can say the genetic material, um, but they are like a combination of the cells also is there. So that is called transformation, conjugation, and transduction. So first of all, we will see transformation. So in bacterial transformation, it was first studied by Griffith in 1928 in Diplococcus pneumoniae. And in the transformation, what happened? DNA from donor cell initiates heritable change in recipient cells. So like here, the bacterium with gene for capsule that is going to be formed some changes into this DNA, and the transformation involves only one trait, through several may be acquired independently in this way. So what happened in this, like when the transformation of the material is done, it means the that is transformed to the other cell. So when bacteria die or when they are reproducing very rapidly, they release fragments of their DNA into their immediate environment, and these DNA fragments are taken to other bacteria by means of diffusion. Like when this DNA is this bacteria is died off and this DNA sheds out, the fragments are just moved into the environment. So some other bacteria can get this DNA, can get this DNA and incorporate it into its DNA. So genes taken and expressed. So here the genetic material can be different now. Now we will see conjugation. Conjugation, it was first reported by Lederberg and Tatum in 1946 in E. coli bacteria. So conjugation takes place only between bacteria of the same or closely related species. So what happens? Cell to cell union occurs between two bacterial cells, and genetic material of bacterial cell goes to an outer cell and lengthwise. So you can see here there is one cell, another cell, normal cell, and here the conjugation occurs like a part of the cell which moves from here, and then it is incorporated into this cell. And now this like DNA is a change here, and now its genetic information is different from the previous one. So for the conjugation, sex pili or pilus forms a conjugating a bridge, as you can see here. This is a bridge like a structure which is formed here with the pili, and the temporary joins with two cells and bacterium that will give DNA is called donor, and the other is called recipient, and that will receive is called recipient. And sometimes it happens that plasmid do this process. Now we will see transduction process. So the transfer of genetic material from one bacterium to another through the third party. That was reported first time in Salmonella typhimurium by Zinder and Lederberg. And in this, genetic material of one bacterial cell goes to another, to other bacterial cell by agency of bacteriophages or phages of viruses or infected bacteria. Like when the virus bacteriophage incorporated in this DNA of virus, and this is of bacterial DNA. So when all the viruses are going to be formed, and this bacterial DNA is just degenerated, or some part of this bacterial DNA is incorporated here with the viral DNA. So they take that DNA, and when it sits on the healthy bacteria, here at this position, so you can say that virus, that bacteriophage transfers that we can say the transformed or transduced DNA into another bacteria and into another its genome. Its genome is now like genetic transform information of the previous bacteria is going to transfer to another bacteria by this third party, that is bacteriophage. Because on the bacteria just viruses can attack, because the viruses are more smaller than this bacteria. So this is actually also the transduction is a sexual method, because of one genetic material from one bacteria, it is going to be transferred or just mingled with the genetic material of the other bacteria. That's why that is called as sexual reproduction, but that is not actually the fusion of sperms and gametes, because they don't produce like this type of gametes. So, dear students, that's it for today. I hope you understood it and just knew and informed about some more concepts about the bacteria, its growth, reproduction, as well as its mode of nutrition. I hope you understood it. Wish you all the best. Allah Hafiz.